we can take very low cost polymers and we can print things that are totally indistinguishable from a cell on these types of instruments. And we can do it cheaply, scalably, consistently, et cetera. So it overcomes all of these problems of supply chain. What the impact of that is, we broaden the percentage of the world that can access this type of control, this type of diagnostic, this type of machinery, et cetera.
Welcome to Business Ninjas, brought to you by WriteForMe, where you'll hear from business leaders who are out there growing their business and slaying it every day. Learn from the masters. Let's get started.
Hi, and welcome to another episode of Business Ninjas. I'm here today with Dr. Jeffrey Kim, founder, CEO, and president of Slingshot Biosciences. How are you doing today, Dr. Kim?
Doing very well. Thanks for having us on the show.
Oh, my pleasure. Appreciate you spending some time with us. Please tell us a little bit about yourself and about Slingshot Biosciences.
Absolutely. So I was originally trained as a scientist in a couple of classical fields, genomics, biophysics, and chemistry, and was introduced to the world of kind of biotech startup life after graduate school, where I did my PhD back in New York, Rockefeller University. I had some friends who were working out in this mysterious place called Silicon Valley, and a couple of startups that were working on some very interesting bodies of work at the time. And I had a bit of a pause before I needed to defend my thesis, and some friends invited me out to come check out the culture, check out the companies. And it was highly addictive. I went into an environment that was growing very rapidly from a biotech perspective, an environment that was very collegiate. We had, at that point, hundreds to thousands of scientists working on the same kind of common goal. And the goal of the company at that time, this was the company I joined Come Out West, was to develop a cheaper, easier, and faster way of making antimalarial medication. So it was also very mission driven. That was kind of my first taste of biotech and industry, I'd say, and I never turned back after that. So being in California was a really opportune, I think, thing to happen. There was a startup culture, obviously, a strong ecosystem of other entrepreneurs who were looking to start businesses to solve problems, and kind of the mantra of, move fast and break things was, very stereotypical. It was very true. People were trying all kinds of interesting business concepts. And about 2 years into my role at the company, because they ended up making me a job offer to come out and diverted me away from academia, it was time at that point to start evaluating some of those ideas. The sector that we had been working in at the company at the time started to pivot, became very challenging. And I figured, hey, why not try this myself? So in around 2012, I left my position at the company and then started two businesses at the same time, which was, in retrospect, extremely reckless and difficult. Doing one startup is tough enough. And I thought, hey, why not hedge my bets and do two? I'm lucky that I was a lot younger than I am today when I made that decision. I was just say that. So that was sort of my first foray into entrepreneurship, and when I started the company, and then from there, the rest is history in some ways. But during that journey, I took a number of interesting segues and side turns, I would say, professionally. Ended up bootstrapping the first company that I started completely. So self-financed, bootstrapped through the entire journey of the business. And then ended up getting acquired at the end of 2017, 2018 timeline. And at that point, I spent some time incorporating the intellectual property team and technology from that business into the acquiring business, and was recruited to work in venture capital as a managing director and a partner at a fund. So I went to the other side of the table for a few years, did some company creation, some minority investment. And at that point, Slingshot had been organically kind of growing. It always been kind of a self-funded, I don't want to say Phoenix rising from the ashes, but it was a little bit ignored during the time because the other company was growing so fast. But what happened during that time is we collected an enormous amount of VOC. We had the fortune of working with the biggest Fortune 50 life science companies from day one, pretty much. And during that time period, we realized that there's an incredible opportunity to create a foundational reagent that everyone in healthcare and biotech would need. But there were some technical hurdles we had to overcome. And during that time of starting, growing, and selling the other business, working in venture capital, we were able to overcome some of those technical barriers, and then Slingshot started to grow very rapidly. And that was the time when I realized it needed, full-time, real card-carrying operating leadership. We needed to bring on institutional capital and grow the business and kind of take it and see how far we could go. And that's what we started doing about 2.5 years ago at this point. So we grew quite rapidly from that point on.
it sounds like you were in the perfect place at the perfect time, just an incredible, fertile atmosphere of like-minded people and enough capital around to move anyone's thoughts forward.
That's correct. We were very fortunate that the origin story for the idea is very classic Silicon Valley, right? We, in a couple of ways, a bunch of coworkers and I were having dinner and drinks one time after work, and we all worked at that point in different settings, different companies, but people were, as everyone does, complaining about their jobs, about the difficulties. And I realized that dinner/drink conversation, that everyone was struggling because of the same problem, which was the problem Slingshot works on. For one reason or another, their clinical trials were being paused or their research was being paused. Or totally sidetracked because they couldn't get access to cells, a consistent source of cells. And during that conversation, I realized that they don't actually need a cell for what they're doing. They just need something that looks and acts like a cell. So you can actually make these things out of something that's non-biological and overcome a lot of the problems that they're facing. And that was the origin story, kind of seed that was planted in my brain. I happened to be thinking about starting a company at that time, so I actually ended up giving my notice at my job like the following day, starting up the company in my basement. So I lived in a live-work loft at the time in West Berkeley, and within 2 weeks already having prototypes shipped off to the first customers. So it was a really fun time.
It seems like some of the greatest businesses and concepts have come out of basements and garages in the last 30, 40 years.
Yeah.
It's a classic story, and I applaud you. You literally gave notice the next day after that dinner.
Yes, it happened to be good timing. It was funny because I told my friends at that dinner that I had done that, and they were panicking because they thought that the dinner conversation pushed me in that direction. But I was ready at that time, so it was just a little push over the edge.
There's different versions of crazy, right? There's
Yeah.
good crazy, and then there's that guy.
Yeah.
So in effect, obviously, every cellular experiment in the world requires a control, and you are now providing that control for most instances. Can you get a little more technical or a little broader for our audience,
Yes.
so they can understand what Slingshot Biosciences is doing that is so revolutionary?
Yeah, I can give a couple of examples that I think will resonate with a broader audience. So I think it's safe to say that pretty much everyone who will listen to this podcast has had a blood test at one point in their lives. If they're born in the US, it's immediately when you're born, and then throughout your lifetime for multiple reasons, whether it's routine, health checks, or if you're sick, et cetera, or donating blood. So there's an enormous volume of blood that is tested. And to test blood, what typically happens is you go into a hospital, they draw a portion of your blood, and then they run it on a machine that analyzes the size, shape, and morphology of your blood. And from those basic features, you can tell whether someone has a malignancy, a disease of the blood that's impacting, or if you have a metabolic disease, sometimes it will impact blood composition and so forth. That process is super high volume. It's run thousands of times a day, actually up to around 700 times a second in the US. There's extremely high volume assay. So when you do that, you actually have to compare the patient's blood to something like a reference, almost like a measuring stick. You can't really measure something without a ruler, for example. So those products are called controls. And those controls ideally are the blood of a healthy human patient, right? You would take someone who's healthy, run it against your blood, and then say, hey, does this look unhealthy? The challenge is that it's very difficult to stabilize human blood. When you try to take a healthy donor and you put their blood in a tube, most of the major populations of blood that you actually want to measure, because blood's actually comprised of many different cell types, they actually disappear, they degrade, they lyse, they go away. So you can't actually build a control out of that material very effectively. So what they figured out many, many years ago, if not decades, is that you can kind of simulate a healthy human blood control with animal blood. And what they started doing is they started spiking in various types of chemically treated animal blood that kind of looks like human blood, and it was slightly more stable. And then fast forward to the world today, and a lot of these controls that you and I have had our blood compared to are made out of donated human blood, but also mixed with things like alligators, sharks, donkeys, geese. So there's a very crazy milieu of animals that are actually required to make routine hospital blood controls that are run millions of times a year. That, for example, is just one example. And the issue with that is the product is still highly unstable. The supply chain is very complex. It drives up the cost. So it makes it inaccessible in many places of the world. It also makes it difficult to draw accurate diagnoses because you have to make many batches of these products, and they're highly variable because it's this crazy mixture of animals. The icing on the cake is that some of those animals are actually now CITES listed, so they're endangered in Europe. So a major critical healthcare product that is required for routine blood diagnosis is at risk, essentially. We make a product that is totally synthetic. We print it using technology leveraged from the semiconductor industry out of polymers. So we can take very low cost polymers and we can print things that are totally indistinguishable from a cell on these types of instruments. And we can do it cheaply, scalably, consistently, et cetera. So it overcomes all of these problems of supply chain. What the impact of that is we can lower the barrier to accessing this type of diagnostic technology because you can't really run a hematology analyzer without a control. And if those controls are expensive, you cannot operate that instrumentation. So what we do is we broaden the percentage of the world that can access this type of control, this type of diagnostic, this type of machinery, et cetera. That's just one example. On the other end of the spectrum, there are cells now being used as advanced therapeutics. For example, CAR Ts or CAR NKs, so engineered cell therapies as they're broadly known. It's an absolutely breakthrough therapeutic modality. The percentage of patients that are permanently cured, and not just treated, but permanently cured is unheard of, which is why the foundational technology won a Nobel Prize recently, and there's so many companies going in that direction. The challenge there is the field is kind of like computers were in the 1940s with vacuum tubes. They showed an amazing POC. They showed something that really works, but it's super hard to manufacture in a scalable fashion. Just like vacuum tubes, they worked, but the power requirements were too high. It was too expensive to manufacture. You couldn't scale it down to build like an iPhone. Fast forward to what we are working on is we've distilled down what an engineered cell therapy does to first principles, and we realized that you don't actually need a cell again. You need something that functionally works like a cell in some regards. So we're able to, and we're working on a fully synthetic form of this class of therapy where we just print the cells. And as you can imagine, it's a heck of a lot easier to print these things than actually draw a patient's blood, genetically engineer it, QC it, and then re-inject it back in, which is the main cost driver and complexity driver of this class of therapy. So that's the other spectrum of how we're replacing blood-based products. So it's both on the routine diagnostic side, which is commodity, and then also in the therapy side.
And I presume that these are stable and they're not air sensitive and there's no shelf life. And on the blood front, when you're talking about sourcing from so many species of animals, there's a public perception. We're in an ESG type of time when people want to invest their money in things that are sustainable and are ethically friendly and things like that. So you kind of tick all the boxes moving forward in terms of sustainability and shelf life and accessibility and public perception of what you're doing.
Yeah, we're really, really pleased about that. Our North Star has always been about access. We want to work on this technology because it improves access to diagnostics. It lowers the cost of therapies. And you hit the nail on the head. One of the major reasons is, shelf life, complexity, cost of manufacturing, et cetera, which makes some of these engineered cell therapies up to $500,000 a dose. It's very, very expensive and very inaccessible, as you can imagine. So by printing them on a product, to your point, that is stable, it's very consistent, very easy to make, we can lower the cost of that by several orders of magnitude and start to work on things outside of advanced cancers, right? Where it's being applied right now.
You're talking about rippling the pool at an exponential level on all fronts. Sorry, the hairs on my arms just stood up because the possible applications of what you're talking about is sort of universal in terms of addressing, viral problems and genetic problems and cancer cells and all sorts of things.
Yeah, that's why I think the business really caught my attention. I had seen in the previous company that I built, the previous companies, plural, we had addressed a very specific problem. What I liked about Slingshot was that it's probably the most ubiquitous thing you could work on is a synthetic cell. And the impact is across every single bucket, whether it's diagnostics, manufacturing, therapeutics, preclinical development, post-clinical development. There is somewhere that we can provide value to either make a process better and faster, or to completely replace that process with a synthetic product that is easier to access. So it's been very satisfying from that regard. One of the challenges we have as a smaller startup is I would love to work on everything, but the way that we have rolled out the business, which I think is the right way, is we focused on near-term revenue generating opportunities because there are many. And then we're going after those North Star programs, but we're doing that in a very conservative manner. The way I've always run my startups is a little bit different. Like, yes, we have taken on institutional capital and venture dollars in this business, but we do it primarily to fuel commercial areas that we know there's revenue coming in.
And the name, I mean, A, it's catchy and very marketable, but B, it signifies that you're able to slay a giant with a small tool. Is that sort of the intent of the name?
Yes, absolutely. That, when we were operating in my basement with, two people originally, that's where the thesis of the name came up from, because we were trying to take down this massive industry that is actually a little cryptic. Not many people know that, for instance, that there are these hidden slaughterhouses for animals that are all over the world. But it's a very large industry, a multi-billion dollar industry. And we were trying to disrupt that as a small 2-person company at the time. And it's satisfying to see it's many years later, but it's really great to see that we are starting to displace a lot of that supply chain, which is really, really great. The other interesting technical part is, which is why I love the name even more, the way we actually make them is kind of like a fluidic slingshot, but a really small one. So that was the other kind of thesis of the name. So it had 2 reasons.
Excellent. Excellent.
Yeah.
Now, is there any competition in your space? Is there anybody else trying to accomplish the same goal?
That's a great question. So when we first started, there were some pretty big competitors, I would say. We were always worried about the huge multinational life science companies that were operating these slaughterhouses. And we were concerned that, the customers or the end customer would be too entrenched or wouldn't be interested in this technology. But the nice thing was that they've all converted over to us. So we had a market map of our risks, basically, of all our competitors when we first started the business. And my co-founders and I, we'd love to go back to that because every one of them is now a customer of ours. So we're selling both into the market directly, but also supplying the legacy providers because they see that this is the future. Because not only does it help with their current supply chain, I can give you a very poignant example for creating and expanding the market. There's tremendous opportunity. So if you are running a clinical trial for a rare disease, for example, in order to measure the effect of your drug, you typically need to have some baseline. You need to have some controls or reference cells. And as you can imagine, getting rare disease control cells is even harder than getting healthy control cells, which is already hard enough. So a lot of times you can't run a clinical trial. So there have been really debilitating diseases that affect millions of people for decades and decades, but no one has actually been able to operationally complete a clinical trial because there's insufficient source of material. We are able to create that rare disease material on demand. Again, we can print these products sometimes in an hour, and that is really opening up that world. So we're working with multiple pharma companies on a constellation of these rare diseases and expanding the total addressable market there. So I would say from legacy competition, they're all our customers. So it's kind of a frenemy type situation where we're displacing their existing supply chain, but they're also buying and they want to displace their existing supply chain. From the, I would say, newer technology standpoint, there are some companies that are peripherally related, but no one that's doing exactly what we are doing. Having said that, I imagine that there will be more competitors. So we are moving very aggressively in the commercial front to kind of build a commercial mode.
That makes so much sense. You can do more than just replicate healthy cells. You can replicate any sort of cells. So you can have the control for any disease or virus as long as there's enough data to replicate it accurately.
Absolutely. That's all we need is the data, and then we can print something and unlock a whole world of the ability to do a trial and whatever you want to develop a diagnostic. There's a lot of really cool things we can do.
Again, I've got hair standing up on my arms. It's fascinating. Who are your customers? What sort of verticals are you servicing at this point?
Yeah, so our original focus with the business was on life science tools and diagnostic companies. So big OEM equipment manufacturers that make some of these instruments. So the hematology analyzers, the flow cytometers. And those instruments typically reside in clinical settings. You can think of a reference lab like LabCorp, Covance, Quest, et cetera, or in major universities or in hospitals. So the original customers were we call them controls and diagnostics or analytical controls for those sectors. What we started to move into more recently is biopharma and cell therapies. As I mentioned, we not only have aspirations of developing a fully synthetic cell that can be the therapy, but we also produce today and sell today a number of very, very helpful products for control manufacturing. So when you go and try to make an engineered cell therapy, there's around 13 or 14 steps that require some form of quality control. It's very important that happens correctly because you're taking a patient's cells, you're isolating and expanding a subset of them, genetically engineering them. You have to QC the heck out of that, obviously. Then you're re-injecting it back into the patient. One of the challenges there is it's the rare disease example times 1,000. It's even harder to find, an engineered cell therapy control intermediate state, for example. So typically what happens is that these companies need to make their own. So they come up with a cocktail that kind of simulates some intermediate manufacturing state, and they use that as their control. That adds significantly to the cost of the drug. Some companies are spending 15, 20, $25 million a year just on making these controls. So what we're doing there and what is growing very rapidly is creating some of these manufacturing controls. In addition, we're also making manufacturing aids. So there are some types of cells that are used to help in the manufacturing of these therapies. So certain cell subtypes that will select and activate and expand the therapeutic subclass that you want. And again, we can make synthetic forms of these that make that process much easier and much more consistent. So we're kind of spreading our tentacles, if you will. But we service the entire healthcare market, I would say, all the way from research through commercialized therapeutic.
So you're not only cutting off problems with the supply chain and giving people access, you're also exponentially lowering the cost of research and development so that one would hope people can have access to medicines that they can actually afford, that they desperately need on the other end of the equation.
Absolutely. That's the North Star. We think that in cell therapies in particular, where I mentioned it can be $500,000 a dose, interestingly, a lot of that price is COGS, so it's cost of goods sold, which is unusual for biopharma. If you think about a small molecule or a biologic, it's actually not very expensive for them to make the drug, just physically make it. But in the case of a CAR T therapy, there are major biopharmas that actually lose money on the therapy because it costs them so much to make it. And there's two effects of that. One, the therapy itself becomes too expensive. And then two, the net effect of that is you can only really deploy that against what's called advanced leukemia and lymphomas in highly developed nations in very specific locations around universities. So it really limits where you can deploy an engineered cell therapy. What we're doing is lowering the cost of their process to manufacture it, but then our North Star will unlock a totally new world where we can lower that cost by orders and orders of magnitude and start to go after things like infectious disease, where it would be just completely economically feasible to go after right now because it's too expensive. So we really want to improve access from the patient side, but then also totally open the gates in terms of how to apply them.
You are rippling the pool in a way that most people dream of. I applaud your efforts.
Thank you.
Are there any other challenges that you're solving for your clients that we haven't covered in our little discussion?
Yeah, I would say we're working on a number of other types of controls, I would say, that could have secondary effects in other markets. So our primary markets have been in liquid analysis. It's mostly been hematology and flow cytometry. We're also expanding into traditional like immunohistochemistry, tissue staining, et cetera. As you can imagine, cells are important there. One area that I am actually very excited about is we're expanding our efforts in the functional cell arena to go after totally new markets. So if you think about what we're trying to do is we're creating a synthetic cell that can either target, attack, and kill a tumor, or it can be a very powerful targeted way of modulating certain immune cells. And as you can imagine, the number of applications there is quite large. It goes beyond let's call them cancers, for example. It goes into the arena of gut microbiome engineering, for example, or general immunomodulation, which has a number of downstream effects. So we do have some research efforts in the microbiome arena. Not prepared to talk about them quite yet, but we can loosely say that there have been very exciting connections made between, for example, the gut-brain axis, behavior, and internal gut microbiome. And we believe we have, again, with our synthetic cells, a really unique platform to start to engineer that in a way that will have physiological effect. So those are other kind of farther out programs that we are playing in.
Excellent.
Yeah.
I'm going to shift gears into sort of a marketing lane for a minute. It is a day and age where we have to tell our story so that people know they should be doing business with us. What role has content played in the growth of Slingshot Biosciences?
I think it's an absolutely critical role in this phase of our business. We were stealth for a really long time, literally in the deep, dark basement for 6-7 years, working a small group of engineers. And then we came out with this technology once we were able to scale it. And we had some really strong customers at first, but then the response initially was, this is too good to be true. This is kind of out of left field. Why has no one done this? So content's been really important to build the brand and the customer realization that A, this is real, B, we're servicing all of your competitors. So why don't you also buy a product from us? And then C, kind of laying the groundwork for like why this is feasible and telling that story in a more scalable fashion. And Caitlin and Marsha, and marketing team at Slingshot, have done a fantastic job of getting that message out. And we're seeing that net impact, because there's only so much you can do if your sales team's going out there and people immediately say, I don't believe you. But if you go out there and content has really prepared them to say, okay, I've seen the data, I've seen the work, I've seen the examples and the case studies, this resonates with what my problem is, let me give this a shot. And we're really seeing this virtuous cycle where content is really helping our revenue, which is great.
Excellent. All right. Fast forward a year from now, what's something you'd like to be celebrating personally and professionally?
Yeah. So professionally, I would love to see our products in more hands, just as many hands as possible. We are on the cusp of having that direct impact on COGS that I mentioned to you for cell therapy. So we're working with some partners that have commercialized cell therapies, some of the biggest and best companies out there. And a really exciting thing to me personally was, when I was younger, I always wanted to help people, right? From a healthcare perspective. My father was a physician, ended up losing my grandfather and several family members to cancer. So when I was a kid, I was like, how do we treat this? And the more that I learned about it, the more daunting it became. So this was part of the thesis as to why I went into research, why I studied chemistry, et cetera, biophysics, was to see, okay, what does the drug development process look like from the very beginning? And how hard is it? And the answer was, it's incredibly hard. It costs billions and billions of dollars. And you may have one or two chances in your lifetime in a biopharma company, because the approval processes are so long, to actually try to launch something that may or may not have a marginal effect in the end on the patient. And that was disorienting. It was like, wow, this is super hard. What I love about Slingshot is that we are selling products today that are actually able to save people's lives. And we're not at the injectable therapy level yet, just to be clear. But to give you an example, and what gets me up, out of bed, is if you look at a vein-to-vein process for a cell therapy, a lot of times that takes between 2 and 4 weeks. And a lot of times they're enrolling the sickest patients that have not responded to any other therapy. In that 2 to 4 weeks, around 30% of the patients don't make it because they're so ill. But of the patients that are treated, a significantly high portion of them survive. So if you can get that drug into the body faster, just saving time, that will save lives today. And that's what we are able to do today. And the reason why is the process for scaling manufacturing, QCing right now takes an enormous amount of time. And it's very difficult to have high quality therapy at the end of that. What our products do is allows that to go faster for lower cost and with more precision. So our customers are using this today, which is great. So it's really satisfying to almost short circuit the life goal of helping patients at scale with this kind of side entrance that we found, which it's just really satisfying. So I would love to see more of that in a year. On a personal level, that's an interesting question to ask an entrepreneur. You kind of lose yourself in the business, and it's hard to think about that. But I would love to see our team, really align around this, and then to see the impacts of it more broadly outside of the company. It's been really satisfying. Again, credit to Caitlin and Marsha and our marketing team to see, friends in my network, acquaintances, people I don't even know, starting to hear about what we're doing and getting excited about it. That's been really satisfying because they can rally behind what we're up to. And that's just a personal goal, is to get more of that message out.
I meet people with lofty goals. You are ticking many of your own goals off as you go through the years, and they're the kind of goals that affect humanity broadly. So again, I thank you for your work, and I wish you all the best in reaching all of those lofty goals that you're talking about. Please tell us what URL people can use to find Slingshot Biosciences, and what social media channels you're using these days.
For sure. The base URL is just slingshotbio, one word, .com. And in terms of social media, we have a presence on LinkedIn. I believe that is primarily it. Yeah, aside from that, I think it's pretty light.
Dr. Jeffrey Kim of Slingshot Biosciences, it has been my honor to have you here today on Business Ninjas. I applaud your efforts. And again, you are affecting humanity a level that most people don't ever get a chance to. So I wish you and
Appreciate that.
your company all the best in the future, because you're going to help a whole lot of people thrive and survive, as it were. So I appreciate what you do. Thank you so much for being with us today.
Thank you very much for your time.